Wishing all viewers and listeners a very pleasant day. The topic of the discussion here is regeneration in invertebrates and vertebrates, as well as development of immune system in vertebrates. We will start with regeneration in invertebrates and vertebrates. Regeneration is an excellent ability of the adult tissue to repair any damage done to the body by autotomy, that is, voluntary separation of a part of the body and accidental injury in nature or imposed by man under experimental conditions. This may involve replacement of dead or damaged cells, healing of a wound, replacement of lost part or the regeneration of the whole organism from a piece of body. It is common in planarians, nemerteans and annelids, but poor in mollusks, nematodes, arthropods and echinoderms. Among vertebrates, it is remarkable in urodels, but limited in anurans, fishes and lizards, and very poor in birds and mammals. Types of regeneration. Common types of regeneration includes number 1, epimorphosis or epimorphic regeneration. This occurs when any part of the body is repaired due to addition of the parts to the remaining piece of that part, as for example, regeneration of amphibian limb. And number 2, morpholexis or morpholactic regeneration. It is a type of repair involving a reorganization of the remaining part of the body of an animal, as for example, regeneration of new individuals from body pieces of hydra, planaria, etc. Other rare types of regeneration includes number 1, heteromorphosis or heteromorphic regeneration. This type of regeneration occurs when a particular organ is amputated. The remaining portion in some rare cases may develop into a different organ. In Palinurus, a shrimp, the eye may regenerate after being removed, but in case the eye is removed along with the optic ganglion, it will not be regarded, and in place of the lost eye, a regeneration blastema is formed, which develops as an antenna-like organ. Number 2, super regeneration. It occurs when the amputated limb or tail is replaced by double or multiple structures. Regeneration in invertebrates. Regeneration in microorganisms and protozoans. Single cell marine algae, Acetabularia, which has 50 mm length and its body part as small as 1/100th of the total size, is capable of regenerating new individuals. Very small segment of stantor and spirostomum are capable of reconstitution, in which ectoplasm plays important role. Experiments showed that when nearly all of the endoplasm of stantor is withdrawn through surface incision, leaving cortex and nucleus, endoplasm is restored eventually, resulting in normal growth. But if the complete cortex is withdrawn off, the cell dies. Regeneration in sponges. The method of regeneration is relatively pronounced in sponges. Complete animal can be reconstituted even from only some undifferentiated archaeocytes. Regeneration in coelentrates. In the coelentrates, the regenerative ability is very high. Hydra, being a lower animal, shows regeneration to an amazing degree. If cut into 2 or more parts, each part reconstitutes into a new and complete individual of reduced size. Hydra shows polarity or anterior-posterior differentiation in regeneration. Thus, the posterior part of a cut hydra regenerates the mouth and tentacles, while the anterior part regenerates foot and adhesive disc. If only the head is split into two, a peculiar two-headed or Y-shaped hydra is formed. Totipotent interstitial cells proliferate and migrate to the damaged end and form blastema, resulting in reorganizing the remaining mass of the body. Other coelentrates regenerate to varying extents, the regenerative power being highest in the polypoid form and much reduced in the medusoid forms. Regeneration in Platyhelminthes. Planarians among Platyhelminthes possess exclusive powers of regeneration. When planarians are cut diagonally, each part of the body will regenerate the missing half. A planarian can regenerate the whole organism from a middle section with the pharynx. If the front end of planarian is cut along the length into two or more parts, every part develops into a new head, resulting in a many-headed planarian. In planarians, control of regeneration is highest near the head and lowest near the posterior end. This is interrelated with the metabolic rate, which is maximum in the head and becomes minimum towards the tail. This variation in metabolic rate is known as metabolic or axial gradient, and such type of regeneration combines epimorphosis and morpholexis. Regeneration in Nemerteans. These animals possess maximum regenerating ability and a complete worm can be formed starting from a very small fragment. Regeneration in nematodes. The regenerating ability is very low, in which only closure of superficial wound is possible by high degree of cell differentiation up to fixed limit of number of cells. Regeneration in annelids. If any oligochaete is cut into two pieces, the posterior one regenerates the frontal end with the mouth and the anterior half regenerates the new posterior end. In Allolobophora, if 5 segments or less are cut off from the posterior end, the regeneration is complete. If the cut is beyond 10 or 14 segments, only 4 or 5 anterior segments are regenerated and the genital organs are thus never renewed. Thus, polychaetes and oligochaetes regenerate anterior and posterior ends after amputation. Among annelids, regeneration is somewhat higher Chaetopteris. The Hirudinea do not regenerate at all. Regeneration in arthropods. In Arthropoda, regeneration is limited to the renewal of the lost part. In most crustaceans, like limb can regenerate at any stage of development, while in insects, limb regeneration occur only in larval stages and regenerated limb do not often reach to normal limb size. Legs of crabs and some spiders regenerate if broken off at second leg joint or if seized by enemy and show autotomy. Regeneration in mollusca. The regeneration is relatively poor in mollusca. In gastropods, eye stalk with eyes as well as parts of the head or the foot may be regenerated. Cephalopods may regenerate their arms, but not the other body parts. Regeneration in echinoderms. The starfishes, brittle star and sea lilies can regenerate arms and parts of the disc. A starfish can regenerate complete organism from just one arm and the central disc. The Holothuria is capable of regenerating the respiratory tract and the alimentary canal, which are frequently ejected through the anus. Regeneration in vertebrates. Regeneration in fishes. In fishes, regeneration power is limited to the fins only. Regeneration in amphibia. In amphibia, regenerative power is highly specialized and most spectacular in urodal amphibians in larval as well as adult stages. Tails, external gills, upper and lower jaws, lens and retina of eye can regenerate. In anurine amphibians, the limbs and tails of tadpoles can regenerate, but adult frog loses this capacity. In newts and salamanders, repair starts in limbs by spreading the epidermis over the wound, followed by closing of wound. Then it bulges out and attains a conical shape by accumulating proliferative cells under the epidermis and is called as the regeneration bud or blastema, which grows and it redifferentiates rapidly in flattened form. Soon, the digits appear, followed by formation of bones and muscles. After several months, regeneration completes and the process is commonly known as epimorphosis. Regeneration in reptiles. When tails of reptiles like lizards are broken off, a new tail regenerates which differs from the original one because of simplified vertebral column and scales of a different type. Regeneration in birds. In birds, regenerative ability is rather poor and only the beak can be regenerated. Regeneration in mammals. In mammals, except few marsupials, regeneration power of limbs is very poor. Tissue regeneration is equivalent to wound healing. Skeletal tissue, tissues of liver and some other internal organs are able to proliferate to a very great extent. Physiological processes involved in regeneration. In the first stage of regeneration, predominantly catabolism, and in the second period, anabolism dominate as far as regenerating animals' physiological state is concerned. Catabolism. This period is also known as destructive metabolic stage, in which there is considerable dedifferentiation of adult tissues at the beginning of regeneration. This period is of greater significance in urodal amphibians. In this stage, abrupt increase in the activity of proteolytic enzymes take place till 11th day, which results into dedifferentiation of parts of the healthy tissues adjacent to the wound surface. Amount of free amino acids is doubled from total nitrogen in normal tissues due to dedifferentiation. The respiratory quotient, that is RQ, in the degenerating tissue falls abruptly, which is a sign of incomplete oxidation in the tissue. Free amino acids, lactic acid and free SH group gets accumulated in the regenerating cells, lowering pH in the tissues of blastema, which slowly rises to normal level. Anabolism. This phase is characterized by an increase in oxidation, increase in RQ, decrease in the amount of free SH groups and of lactic acid, richness of cytoplasmic RNA, increase in size of nuclei in cells participating in regeneration, thus also known as the constructive stage of metabolism. We would now learn the development of immune system in vertebrates. Immunity is the state of protection from infectious disease. It has both non-specific and specific components in vertebrates. Innate or non-specific immunity refers to the basic resistance to disease that a species possesses. It comprises 4 types of defensive barriers: anatomic, physiologic, endocytic and phagocytic, as well as inflammatory. Anatomic barriers. The epithelial surfaces form a physical barrier that is impermeable to most infectious agents, acting as the first line of defense against invading organisms. In the gastrointestinal and respiratory tract, movement due to peristalsis or cilia helps remove infectious agents. Mucus traps infectious agents. The flushing action of tears and saliva helps prevent infection of the eyes and mouth. Physiologic barriers. Physiologic barriers include temperature, pH, oxygen tension and various soluble factors. Many species are not susceptible to diseases because their body temperature inhibits pathogen growth. Chickens, for example, display innate immunity to anthrax because their high body temperature inhibits the growth of pathogen. Gastric acidity also provides an innate physiologic barrier to infection because very few ingested microorganisms can survive the low pH of the stomach. A variety of soluble factors can also contribute to non-specific immunity. These are: number 1, lysozyme. A hydrolytic enzyme found in mucus secretions is able to cleave the peptidoglycan layer of the bacterial cell wall. Number 2, interferons, a group of proteins produced by virus infected cells. Among their many functions is the ability to bind to nearby cells and induce a generalized antiviral state. Number 3, complement. It is a group of serum proteins that circulate in an inactive proenzyme state. These proteins convert the inactive proenzymes into active enzymes. Endocytic and phagocytic barriers. Another important innate defense mechanism is the ingestion of extracellular macromolecules and particles through endocytosis and phagocytosis respectively. In endocytosis, the macromolecules contained within the extracellular tissue fluid are internalized by cells. This internalization occurs as small regions of the plasma membrane invaginate or fold inward, forming small endocytic vesicles. Phagocytosis involves the ingestion of particulate material including whole pathogenic microorganisms. In phagocytosis, the plasma membrane expands around the particulate material to form large vesicles called phagosomes. Once particulate material is ingested into phagosomes, the phagosomes fuse with lysosomes and the ingested material is then digested in the endocytic processing pathway by a process similar to that seen in endocytosis. Inflammation. Inflammation is one of the first responses of the immune system to infection or irritation. Inflammation is stimulated by chemical factors released by injured cells and serves to establish a physical barrier against the spread of infection and to promote healing of any damaged tissue following the clearance of pathogens. The process of inflammation is initiated by cells already present in all tissues, mainly resident macrophages, dendritic cells, histiocytes, Kupffer cells and mastocytes. Chemical factors produced during inflammation that are histamine, bradykinin, serotonin, leukotrienes and prostaglandins attract phagocytes, especially neutrophils. Neutrophils then trigger other parts of the immune system by releasing factors that summon other leukocytes and lymphocytes. Cytokines produced by macrophages and other cells of the innate immune system mediate the inflammatory response. The acquired immune system or the specific immune system is composed of highly specialized systemic cells and processes that eliminate or prevent pathogen growth. One of the two main immunity strategies found in vertebrates is acquired immunity that creates immunological memory after an initial response to a specific pathogen, leading to an enhanced response to subsequent encounters with that same pathogen. In acquired immunity, pathogen specific receptors are acquired during the lifetime of the organism. The cells of the immune system. B lymphocytes. B cells marked by CD45 plus membrane molecules are the major cells involved in the creation of antibodies. Antibodies, also known as immunoglobulin, that is Ig, are large Y-shaped proteins used by the immune system to identify and neutralize foreign objects. In mammals, there are 5 types of immunoglobulin: IgA, IgD, IgE, IgG and IgM, differing in biological properties, and each has evolved to handle different kinds of antigens. B cells express a unique B cell receptor BCR, an immobilized antibody molecule. All the BCR of any one clone of B cells recognizes and binds to only one particular antigen. Once a B cell encounters its cognate or specific antigen and receives additional signals from a helper T cell, it further differentiates into an effector cell known as a plasma cell. Plasma cells secrete antibodies. These antibodies bind to antigens, making them easier targets for phagocytes and trigger the complement cascade. B cell mature in bursa in birds and in bone marrow in other animals. Antigen presenting cells. Acquired immunity relies on the capacity of immune cells to distinguish between the body's own cells and unwanted invaders. The host cells express self antigens. These antigens are different from those on the surface of bacteria or on the surface of virus infected host cells that are non-self or foreign antigens. The acquired immune response is triggered by recognizing foreign antigen in the cellular context of a danger activated dendritic cell. With the exception of non-nucleated cells, including erythrocytes, all cells are capable of presenting antigen through the function of major histocompatibility complex, that is MHC molecules. Some cells are specially equipped to present antigen and to prime naive T cells. Dendritic cells and B cells and to a lesser extent macrophages are equipped with special co-stimulatory ligands recognized by co-stimulatory receptors on T cells and are termed professional antigen presenting cells, that is APC. T lymphocytes. T lymphocytes arise from hematopoietic stem cells in the bone marrow and migrate to the thymus gland to mature. During its maturation within the thymus, the T cell comes to express a unique antigen binding receptor on its membrane called the T cell receptor. Naive cytotoxic T cells are activated when the T cell receptor, that is TCR, strongly interacts with a peptide bound MHC class I molecule. This affinity depends on the type and orientation of the antigen or MHC complex. Once activated, the CTL undergoes a process called clonal expansion, in which it gains functionality and divides rapidly to produce an army of armed effector cells. Activated CTL will then travel throughout the body in search of cells bearing that unique MHC class I plus peptide. When exposed to these infected or dysfunctional somatic cells, effector CTL release perforin and granulysin, that are cytotoxins that form pores in the target cell's plasma membrane, allowing ions and water to flow into the infected cell and causing it to burst or lyse. CTL release granzyme, a serine protease that enters cells via pores to induce apoptosis. Upon resolution of the infection, most of the effector cells will die and be cleared away by phagocytes, but a few of these cells will be retained as memory cells. There are two subpopulations of T cells, T helper, that is Th cells, and T cytotoxic, that is Tc cells. T cytotoxic cells, also known as Tc, killer T cell, cytotoxic T lymphocyte or CD8 plus cells, are a subgroup of T cells that induce the death of cells that are infected with viruses and other pathogens or are otherwise damaged or dysfunctional. Helper T cells or CD4 plus lymphocytes are immune response mediators and play an important role in establishing and maximizing the capabilities of the acquired immune response. These cells have no cytotoxic or phagocytic activity and cannot kill infected cells or clear pathogens, but manage the immune response by directing other cells to perform these tasks. Helper T cells express T cell receptors TCR that recognize antigen bound to class II MHC molecules. The activation of a naive helper T cell causes it to release cytokines, which influences the activity of many cell types, including the APC. HIV, that is human immunodeficiency virus, is able to subvert the immune system by attacking the CD4 plus T cells. Immunization. Immunization, commonly referred to as vaccination, is the deliberate induction of an immune response and represents the single most effective manipulation of the immune system. Immunization is successful as it utilizes the immune system's natural specificity as well as its inducibility. Immunological diversity. Most large molecules, including virtually all proteins and many polysaccharides, can serve as antigens. The parts of an antigen that interact with an antibody molecule or a lymphocyte receptor are called epitopes or antigenic determinants. At the end, we would conclude the topic that we have talked so far. The regeneration process is comparatively higher in lower animals. In general, it may be related to high degree of differentiation in higher animals. Hence, in vertebrates it is limited to wound healing or epimorphosis. This process involves amputation, differentiation, blastema and redifferentiation to form lost parts of the animal. The physiological processes involved are catabolism and anabolism. The immune system develops into two ways, that is, innate and acquired types. Innate immunity is present by birth and acquired immunity is complex and develops later. Acquired immunity is dominant in all vertebrates against all types of foreign antigens for protection. Immune reactions are well organized by the cooperation of innate immunity with acquired types in vertebrates.